Internal or tissue respiration refers to the gas exchange that is happening between blood and tissue cells. So when blood flows through the systemic blood capillaries, that's where we expect oxygen from blood diffuse into the tissues. Basically, tissue cells get oxygen, and carbon dioxide that is produced by tissue cells should diffuse into blood, so blood can take carbon dioxide away from the tissues, and eventually we know carbon dioxide must be eliminated. We already know that there are two components of blood that both transport oxygen and carbon dioxide. Most of oxygen is transported by erythrocytes, red blood cells, and a very small percentage of oxygen is in plasma as dissolved gas. When it comes to carbon dioxide, we see that about 23% will be transported by red blood cell, and 77% of carbon dioxide is in plasma. The only difference is that 7% is dissolved in plasma as gas, 70% travels in the form of this important buffer, which is bicarbonate ion. So let's quickly visualize inside a red blood cell, an erythrocyte. We know that inside the red blood cell we have about 280 to 300 million hemoglobin molecules. The maximum capacity of hemoglobin for oxygen would be 4. When 4 oxygen molecules are attached to a hemoglobin, we name this oxyhemoglobin. In videos and physiology textbook, usually that's how we show oxyhemoglobin. When red blood cells get close to the tissue cells, usually each hemoglobin is willing to let go of one of its oxygens. Hemoglobin is not going to release all its oxygen molecules. So in most cases, only one oxygen molecule separates from hemoglobin. Please note that this is like a business transaction. For this hemoglobin to let go of one of its oxygen, it has to receive something. If tissue is highly active, let's say you're exercising, your skeletal muscles need extra oxygen. If they're willing to, for example, give hemoglobin 2 carbon dioxide molecules, then hemoglobin would be willing to let go of 2 oxygen molecules. But that's the max that hemoglobin is going to break down its bond with oxygen and let go of oxygen. As soon as hemoglobin is attached to less than 4 oxygen, we change the name and we name this deoxyhemoglobin. But please keep in mind that hemoglobin is stable when it's attached to 4 things. So if we have 3 oxygen molecules attached to hemoglobin, something else must be attached to hemoglobin in order to stabilize this compound. And in deoxyhemoglobin, we're trying to figure it out. What are those things that could bind to hemoglobin and convince hemoglobin to break its bond with oxygen and release oxygen to the tissue? So here we have a view of red blood cell. First, the red blood cell is inside a pulmonary capillary, inside lungs, and we see that the bond between hemoglobin and oxygen molecules is very strong and stable. The reason is attraction between hemoglobin and oxygen depends mostly on the partial pressure of oxygen. Please keep in mind, when we say partial pressure, it simply means the pressure of that specific gas in the mixture. When air gets inside the lungs, we need to acknowledge that air is not just pure oxygen. Air is a mix of different gases, and the pressure of air would be the combined pressure of all these gases. In pulmonary capillaries, when we focus on pressure of only oxygen, we notice that this pressure is really high. It's around 100 millimeter of mercury. When pressure of oxygen is so high, the attraction between hemoglobin and oxygen is also very high. Hemoglobin forms bond with oxygen and is not willing to break the bond. But as soon as we get inside the systemic capillaries, those capillaries that we have close to our tissue cells, and we know that tissue cells keep using oxygen, so it's not surprising that the pressure of oxygen in systemic capillaries is low. In a place that pressure of oxygen is so low, we notice that the bond between oxygen and hemoglobin becomes weak and unstable. So this is the moment that hemoglobin is willing to break this bond and release oxygen. However, it needs an excuse. By excuse, I mean hemoglobin has to bind to something in order to break its bond with oxygen. So we have a closer view of the red blood cell inside the systemic capillary, And we already know because pressure of oxygen in systemic capillary is low, the bond between hemoglobin and oxygen is unstable and weak. And we already know that tissue cells produce carbon dioxide through different chemical reactions that they have, including aerobic cellular respiration. So in tissues, pressure of carbon dioxide is high. That explains why carbon dioxide from tissues diffuses into the blood capillary. And we notice that when carbon dioxide diffuses into blood, about 7% of this gas stays in plasma as dissolved gas. 93% diffuses inside the red blood cell. Out of this 93% of carbon dioxide that gets inside the red blood cell, about 23% react with hemoglobin. Basically, carbon dioxide binds to hemoglobin. That would be the excuse that hemoglobin was waiting for. So as soon as hemoglobin binds to carbon dioxide, it breaks the bond that it has with one of its oxygen molecules and releases oxygen. And we know that oxygen quickly diffuses from the systemic capillary into the tissue cells. So in short, we can say the more carbon dioxide tissue cells produce, the more oxygen they're going to receive. The question is, what happens to the other 70% of carbon dioxide that is inside the red blood cell? Inside the red blood cell, we have enzymes that allow this high percentage of carbon dioxide to react with water. The result would be carbonic acid. Carbonic acid quickly dissociates into two ions, hydrogen ion and bicarbonate ion. Bicarbonate ion is the buffer, and we see that most of the bicarbonate ion leaves the red blood cell and goes into plasma. For a moment, visualize what's going on here. The buffer went into plasma. Inside the red blood cells start becoming acidic. The pH inside the red blood cell starts going down. So someone has to save red blood cell, because if inside the cell becomes too acidic, that leads to the death of this cell. Here we're looking for a hero, someone who can act as a buffer. And we already know that hemoglobin is no longer attracted to oxygen because pressure of oxygen is very low in systemic capillary. So hemoglobin basically acts as the hero of this story. Since it's looking for that excuse to break its bond with oxygen, what it does, quickly it reacts with hydrogen ion, and as you see, as soon as hydrogen ion binds to hemoglobin, it no longer affects the pH inside the red blood cell. That's why we say hemoglobin acts as a buffer, and because hemoglobin cannot hold on to 5 things, as soon as it binds to hydrogen ion, it has to release oxygen. So now oxygen is free, it can go to tissue. So that would be another excuse that allows hemoglobin to break its bond to oxygen, and as a result, tissue gets oxygen. Now we see that there are some other oxyhemoglobins still left inside the red blood cell. The question is, what would be another excuse for hemoglobin to release the oxygen into the tissue? The other excuse would be a molecule that is called bisphosphoglycerate. This is a molecule that red blood cell produces when red blood cell does a process such as anaerobic glycolysis. Recall, red blood cell does not have mitochondria, so to generate ATP and do all these different things that this cell does, it has to produce ATP through a process that does not require oxygen, and that would be anaerobic glycolysis. So in short, bisphosphoglycerate is a byproduct of anaerobic glycolysis. When we are inside the systemic capillaries, basically attraction between hemoglobin and oxygen is low. That's the moment that hemoglobin is willing to bind to bisphosphoglycerate, and as soon as it binds to bisphosphoglycerate, it can release oxygen to the tissues. So these are the reactions that show what would be that something else that can bind to hemoglobin and help hemoglobin to release oxygen to the tissue. In short, one would be carbon dioxide, the other one would be hydrogen ion, and the next one would be bisphosphoglycerate. And the more of these 3 that we have, the more tissue is going to get oxygen. And as you see here, now inside this red blood cell, instead of oxyhemoglobin, we have millions of different types of deoxyhemoglobin. We name the deoxyhemoglobin that carbon dioxide is attached to hemoglobin. Please note that there are 3 oxygens still attached to this hemoglobin. We name this carbaminohemoglobin. So we have three different types of deoxyhemoglobin inside this red blood cell. I would like to emphasize a factor, and that would be temperature. Within limits, when temperature rises, these reactions that we see here, which all of them leads to release of oxygen, are going to happen much faster. So that makes sense in those tissues that temperature rises. Imagine, for example, you're exercising, and we know that when skeletal muscles contract, they do more aerobic cellular respiration, and one of the products of aerobic cellular respiration is heat. So in general, when the cells are more active, the temperature of that tissue rises, and those are the cells that are going to receive more oxygen. A simple explanation is that when you increase the temperature within limits, you're speeding up these chemical reactions that lead to release of oxygen to the tissue. So now this red blood cell is ready to go back to the lungs, because we know when we get inside the lungs, the hemoglobin is very much attracted to oxygen, binds to oxygen, and as soon as hemoglobin binds to oxygen, obviously it has to release anything else that was attached to it. For example, this is the place that hemoglobin binds to oxygen, releases carbon dioxide, releases hydrogen ion, and releases bisphosphoglycerate, which in a separate video named external or pulmonary respiration, I walk you through the reactions that are happening inside the red blood cells when red blood cells are inside the pulmonary capillaries. Hope you find this helpful.